Good day, everyone. My name is Yoichiro Kanno, Associate Professor at Colorado State University. My talk today is about the importance of small tributaries to maintain trout populations, and it is based on my research on Japanese salmonids. However, they have much in common with brook trout in terms of their ecology and conservation challenges. So my goal today is to use my research in Japan as a springboard to think about brook trout conservation in the USA. Here's a little bit about me. I'm from Japan. I have a BA in International Environmental Law and an MS from Dalhousie University, Nova Scotia, where I first encountered brook trout. I then worked as a diplomat for the government of Japan for 8 years before coming to the University of Connecticut for my PhD working on brook trout. Then I held 2 research positions at Tennessee Tech and University of Massachusetts before starting my professorship at Clemson University, South Carolina. In essence, I followed where brook trout occur in the East, although I did not plan on that. Since 2017, I have been at Colorado State University, where people consider brook trout a nuisance species as far as native cutthroat trout conservation goes. I want to highlight ecological similarities between brook trout and Japanese salmonids over the next few slides. This map shows historic brook trout distribution in brown and current distribution in green in the USA. I like this map because it acknowledges that brook trout do occur in Canada. Brook trout populations have declined in the USA, and they most typically occupy small headwater streams, and this pattern is most evident in the southern range. And it is fair to say that brook trout is an inhabitant of small streams in headwaters. For example, here's a flyer of our Lunch and Learn session. I did not pick this habitat photo, but this is a kind of stream that people think of as habitat for native brook trout. Conservation effort often targets this type of small streams, whether it is habitat restoration, riparian revegetation, or culvert improvement to improve access. Japanese salmonids also occupy small headwater streams. The photo on the left shows 2 species that we study in Japan, white-spotted charr and red-spotted masu salmon. Photos on the right show our study site, Subsawa Creek. Genetically pure populations of native salmonids are in small, isolated habitats because of habitat degradation, non-native species such as brown and rainbow trout, and hybridization with stocked fish farther downstream. Now, we all know that habitat fragmentation is a key threat to native trout population persistence, and I cannot discuss Japanese native trout conservation without touching upon habitat fragmentation. And our native salmonids occur in really small habitat patches. Here's a work by my colleague Morita and Yamamoto, who surveyed anthropogenically isolated populations of white-spotted charr and found that populations are more likely to disappear in small habitat patches and when isolation time increases, which is a number of years since barriers were built. This figure shows watershed area on the y-axis and probability of occurrence on the y-axis. And different curves are the number of years since barriers were built. Now, I want to draw your attention to the range of watershed area that white-spotted charr occupy. They occupy very small watershed areas, 10 square kilometers or less, which is equivalent to 3.9 square miles, similar to or smaller than native brook trout habitat in the USA. And brook trout and Japanese salmonids, particularly white-spotted charr, are also taxonomically similar. Both species belong to the genus Salvelinus. And here's a story about Dr. Bob Behnke, whom many of you probably know for his articles in the Trout magazine over many years. Dr. Behnke served in the Korean War, and when he got a break there, he decides to go to Japan for fishing. Here on the lower right photo, you see Dr. Behnke holding a rifle and a fishing pole. And once in Japan, he was fascinated that how white-spotted charr looks similar to brook trout that he caught as a child in Connecticut. And of course, brook trout and white-spotted charr both require clean, cold water, so they are also ecologically similar as well. Now, with that background, I will switch a gear and talk about our long-term study that demonstrates the critical role small tributaries play to maintain trout populations. Our work has recently been published, and I encourage interested listeners to find technical details in these two papers. And first, I would like to acknowledge my colleagues who have been conducting this study for 15 years, and it is counting. And suffices to say that it takes a village to do good science. And I will take you across the Pacific Ocean to central Japan, where the study site Sabusawa Creek is located. We have been studying sympatric populations of white-spotted charr and red-spotted masu salmon in Sabusawa Creek. They are landlocked in our study area, although anadromous forms exist at northern latitudes, similar to brook trout. We chose to conduct a long-term study in Sabusawa Creek because it was the last remaining stream in the prefecture, equivalent to a state in the USA, where the two species still coexist. And Sabusawa Creek is also unique in the sense that populations of the native salmonids have persisted despite severe habitat fragmentation. This map shows how severely fragmented their habitat is. Note the scale bar on the lower left. Solid horizontal lines on the map are complete barriers to movement, and one dashed line between section F and G is a seasonal barrier. Basically, there is a barrier every 100 meters, and still charr and salmon populations have persisted for more than 30 years since barrier construction, with charr occupying upstream and salmon occupying downstream, with sections of sympatry in the middle through sections C through E. So we wanted to answer this simple question: why do populations persist in this highly fragmented habitat? And our gut feeling as field biologists was that these two small tributaries shown in red circles were the reasons why the reasons because there were typically more fish in the tributaries than in the mainstem, but fish density was not statistically significant. So our educated guess was that small tributaries were important demographically in terms of fish survival and growth, but we need to take a deeper look at it. And we weren't really convinced of the importance of tributaries when we started the study because these tributaries are very small, about 2 meters wide. T1, the first tributary, is actually 1.7 meter wide, and T2 is 2.1 meter wide. And for the remainder of my talk, I will define 3 habitat patches for each species, which are tributaries, mainstem section with connectivity to tributaries, or M subcon, and mainstem fragmented section that are occupied by both species. We conducted annual mark recapture sampling in October for 9 years, and it always took place in the 3rd weekend of October. October coincides with their spawning season, so we can determine maturation status of individuals. Our dataset included over 1,300 individuals in each species, and we identified individuals by marking fish in the head with different color combinations of VIE tags. So simply put, we basically tattoo fish head for unique identification of individuals. Using field data, we developed a demographic model by following individuals through life stages. Shown here is what's called a life cycle graph, composed of young of the year fish, YOI or YOI, immature, and mature fish. Survival is shown by Greek letter phi, and maturation is shown by omega. We estimated these parameters in each habitat patch, namely tributaries, mainstem connected, mainstem fragmented, to understand spatial patterns of demography and also to infer movement rates between patches. So there's a lot of stats and math going on in the background. And what did we find? We found that survival is higher in tributaries relative to the mainstem. This figure shows mean and 95% credible intervals of annual survival in charr by life stage, young of year, immature, and mature, and by habitat patch, tributary, mainstem connected, and mainstem fragmented. And you can see that survival is higher in the tributary, particularly in the mature fish. And survival is similarly higher in the tributary in masu salmon, although the result is not shown here. However, body growth is higher in the mainstem. Here you see fork length of charr on the x-axis and annual growth on the y-axis. Colors refer to movement patterns where M to M refers to individuals captured in the mainstem in 1 year, again captured in the mainstem in the following year. Now compare orange, which is the growth of mainstem residents, and green, which is the growth of tributary residents, where you see that spatial variation in growth is particularly evident in younger year fish. This pattern once again holds in masu salmon. And finally, fish movement was limited, but when it occurred, it was predominantly in the downstream direction, circled here by red. And notably, movement was asymmetrical from tributaries to the mainstem, and it was as high as 30% in the case of masu salmon moving from the tributary to the mainstem connected patch shown on the right panel. To summarize, survival was higher in tributaries, which explains maybe why there were typically more fish in the tributaries. But growth was higher in the mainstem. And we need to take into account that larger females are also more fecund, meaning that larger females have more eggs. And finally, movement was asymmetrical from tributaries to the mainstem. Now I want to emphasize that all life stages and reproduction occur in both tributaries and the mainstems in both species. Okay. And perhaps these results show why spatial ecology matters in trout. Demographic rates such as survival, growth, and reproduction cannot always be maximized in the same locations. Therefore, fish need to move in the riverscape, and as practitioners, we need to manage for habitat connectivity. But empirical data to support this concept are still few, and our study is one of the few to demonstrate this concept. So let's revisit our research question. Our goal was to understand why trout populations persist in Sub-Salar Creek, and specifically if small tributaries are important demographically. We wished that answering this question had been more clear-cut, but it is hard to tell which habitat was more important based on evidence so far. To understand which habitat patch is more important, we put demographic information together for analysis of population growth rates. So this is similar what human demographers do. Once there is information on birth, mortality rates, we can project whether and how fast human populations grow in our society. And we can do the same for trout. And on the next slide, you will see values of population growth rates, or lambda. So I want to condition you on that slide. Now, if you see lambda is over 1, that means that population is increasing. For example, lambda of 1.2 means that the population grows by 20% annually. When lambda is 1, population size does not change. When lambda is less than 1, the population is decreasing. Lambda was calculated for each habitat patch in 2 ways. One before accounting for emigration, meaning that we let all immigrants to stay in their local patch. And the other where we let them emigrate, which is actually what happened in our data. And we call them pre-lambda and post-lambda in my next slide. And here is mean and 95% credible intervals of pre and post-lambda for each species by habitat patch. Now, pre-lambda, where we let emigrants to stay in their local patch, was significantly larger than 1 only in the tributaries. Now, pre-lambda values in tributaries are 1.2 in both species, meaning that their population growth rates are about 20% if all fish stayed in that tributaries. In addition, the difference between the pre and post lambda is large in tributaries because individuals moved from the tributaries to the mainstem and made demographic contributions there. We don't see these patterns in the mainstem. Therefore, this is the most direct evidence to show that tributaries were source populations in both charr and salmon. So why do the tributaries contribute to demography so disproportionately? In other words, why are the tributaries so important? And here in Sub-Sour Creek, temperature was nearly identical between the tributaries and mainstem, so we can count that out. And we think that this is due to physical habitat complexity. So one year, we brought in this river engineer shown on the left, who had a rod and velocity meter. And he took a bunch of habitat measurements, and we found that there's more wood in the tributary, and velocity was slower in the tributaries. In fact, there were many negative velocity values indicating that water was moving from downstream to upstream because of back eddies. Now, these are exactly the type of habitat features that charr like. And in fact, the Japanese name for charr is rockfish because they love those space between rocks where velocity slows down. And in sum, our research shows that tributaries are small, but demographically mighty. It is notable that tributaries occupy only 12 to 18% of habitat per surface area in these spatially structured populations. So what are the management implications? And I think we're going to discuss further about these management implications in our Lunch and Learn session. Our finding that small tributaries are important for Japanese salmonids has global consequences, including brook trout. As I mentioned earlier, brook trout have much in common with Japanese salmonids, ecologically and taxonomically. But the kind of empirical data to demonstrate the importance of small tributaries are scarce, and this is where scientific contributions comes. Our finding is also important because we continue to debate whether Our finding is also important because we continue to debate where the river begins in US Clean Water Act and elsewhere. And finally, this is a case study of fisheries management in a stream named Zako River in Japan, about 100 miles north of Sub-Sour Creek. They are managing for a thriving white-spotted charr fishery by protecting tributaries. Here on the map, pink zones show no fishing zone, and blue shows fishing zone. So you can see that on the left side of the map, they thoroughly protected tributaries. And on the right-hand side, they did not protect tributaries. And by enforcing this regulation, there are a lot more fish on the left side of the map, and anglers are happy. So I also want to introduce another Japanese phrase called mother seed creeks to refer to tributaries. Now we don't know if this phrase was coined by fisheries managers or anglers, but then anglers are familiar with the word or phrase and they buy in this concept. So our data show that conservation actions to protect tributaries are important. These actions may include habitat restoration, riparian reforestation, non-native trout removal, habitat connectivity, fishing regulations, and climate refugia, which many of the listeners are probably working on. And finally, we also should not discount the mainstem habitat, as there are some recent studies that demonstrate the importance of mainstem for growth and movement in salmonids. And this really embraces the riverscape concept that habitat heterogeneity and complementarity is important in the stream network for trout populations to persist. This concludes my talk. Thank you very much for listening, and I look forward to Lunch and Learn discussion on March 31st.